Welcome! Today, I will be discussing the complete step-by-step process of the major cross-matching procedure, specifically the broad-spectrum compatibility test. The primary purpose of a major cross-match is to ensure compatibility between the donor's red blood cells and the recipient's plasma. As a single drop of mismatched blood triggers an immediate immune system attack, known as an acute hemolytic transfusion reaction, the recipient's antibodies identify the donor cells as foreign, causing them to rapidly swell and burst. To prevent this lethal interaction, the laboratory uses the major serological crossmatch. This procedure is our final defense to detect incompatibilities before donor blood ever reaches the patient. Before handling any biological specimens, we must secure our own safety. Every human sample is a potential source of blood-borne pathogens, like HIV or hepatitis B. Standard precautions require strict barrier protection. Before touching a single tube, the technologist must wear a fully buttoned, fluid-resistant gown, non-latex gloves, and full facial protection. These barriers protect the scientist from the specimen, while the rigid discipline of the cross-match procedure protects the patient from the donor blood. The first critical step is specimen preparation and labeling. Clerical errors at the bedside, specifically misidentifying a patient, are the leading cause of fatal hemolytic transfusion reactions. Labeling requirements are absolute. The specimen must display at least 2 unique identifiers collected directly from the patient's physical wristband at the time of phlebotomy. Once the sample reaches the laboratory, it must be inspected for hemolysis. We must reject any samples where the cells have already burst, as this makes it impossible to detect procedure-induced cell destruction later. Before proceeding to the cross-match, we must always perform an EBO-RH typing verification on both the donor unit and recipient sample. This is the most frequently performed test in the blood bank and acts as our first line of defense. To perform our test panel, we need 2 specific components prepared from the donor sample: a 2 to 5% suspension with the donor red blood cells and a tube of their separated serum or plasma. Isolating the red cells from the serum allows us to test the donor's physical blood antigens independently from their circulating antibodies. This physical separation is the foundation for avoiding catastrophic cross-matching errors later on. We begin with forward typing. The purpose here is to detect the actual physical antigens attached directly to the donor's red blood cell membranes. Set up and label 2 test tubes A and B. Add exactly 2 drops of commercial anti-A reagent to tube A and 2 drops of anti-B to tube B. Then add 1 drop of your prepared 2 to 5% red cell suspension to each tube. Mix the tubes gently to combine the reagents and the cells. Then, place them in a centrifuge for exactly 20 seconds. Afterward, gently agitate the tubes to dislodge the cell button that forms at the bottom. Now we evaluate the results and look at the left side of this split screen. The solid clumping you see is of strong positive reaction, meaning the specific antigen is present. On the right, the smooth cloudy fluid indicates a negative reaction. Reading the specific surface antigens provide the first half of the donor's immunologic profile, telling us exactly what their red blood cells carry. With the red cells typed, we move to reverse typing. This process detects the expected antibodies naturally circulating in the donor's serum. It acts as a mandatory safety check against the forward type. Label 2 new tubes A and B. This time, place 2 drops of the donor serum into each tube. Add 1 drop of known 5% A red cells to tube A and 1 drop of 5% B red cells to tube B. Centrifuge these 2 tubes for 1 minute. After spinning, perform a visual check of the supernatant fluid at the top of the tube before you shake or dislodge the bottle. If you observe a red tint or clear pink fluid, this indicates hemolysis. In blood bank serology, hemolysis means a positive reaction, confirming antibody presence, not a failed or broken test. When dislodging the button, avoid aggressive shaking, as that can destroy weak agglutination. If the reaction is questionable, let the tubes rest for 5 minutes at room temperature to enhance weak interactions.
Landsteiner's rule, individuals naturally produce antibodies against ABO antigens they lack. A person with A antigens on their cells will have anti-B in their serum. The reverse test confirms this inverse relationship, serving as a life-saving cross-check for our initial forward results. Next is the antigen test. This will classify the donor unit as either Rh positive or Rh negative. In a small test tube, mix 1 drop of anti-D typing sera with 1 drop of donor blood. Incubate this mixture at 37 degrees Celsius for 2 to 4 minutes. Then centrifuge the tube at 1000 RPM for 1 minute, adding a drop of saline to aid the reading process. Gently dislodge the cells. Immediate macroscopic agglutination confirms the unit is Rh positive. If you see a smooth negative result, we cannot stop here. A negative reading mandates immediate secondary testing. The D antigen is highly immunogenic. If a D negative patient receives D positive blood, their immune system will aggressively attack the foreign cells. That makes accurate Rh typing just as critical to patient survival as ABO grouping. For samples that initially appear Rh negative, we must execute the weak D protocol. This test is designed specifically to catch dangerously weak D antigen expression that standard testing misses. Looking at this procedure guide, we start by labeling 2 tubes, D and control. Place 1 drop of your cell suspension in each. Add anti-D to the test tube and your Rh control reagent to the other. Step 3 is critical. Incubate both tubes for 15 to 30 minutes at 37 degrees Celsius to allow binding. As step 4 indicates, you must wash the cells 3 separate times with normal saline, fully decanting the fluid each time to remove unbound proteins. Then add your antiglobulin or anti-IgG reagent to the dry cell buttons. Mix the tubes and centrifuge them at 3400 RPM for 15 seconds. Gently resuspend the buttons and examine closely for any sign of agglutination. We end with a mandatory validation check. If your result is negative, you must add an IgG-coded Coombs control cells to the tube. Spin it again and verify that those control cells successfully agglutinate. The failure parameters are strict. If Coombs control cells fail to clump, or if your decontrolled tube shows a positive reaction, meaning the patient cells were coated already with antibodies, the entire weak D test is invalid and must be investigated. If weak D test is positive and the control is negative, the unit is labeled as Rh positive for blood donation. Catching this weak expression prevents a severe hidden immune response from occurring in an Rh negative recipient. We now bring all 4 test tubes together: forward, reverse, Rh, and weak D. This gives us the complete serologic picture of the donor. First, we confirm our logic checks. The forward typing results must match the reverse results perfectly. Then, we verify the weak D test is definitively negative and backed by successful Coombs control validation. With the data aligned, you can authorize the unit.
Next, we prepare a 5% red cell suspension of the donor's red cells.
Step 1 is preparing the donor cell suspension. Pipette a small amount of donor whole blood into a test tube and add a heavy stream of physiologic saline. Spin the tube in the centrifuge at 1000 to 2000 RPM for 1 minute. When it finishes, you will see a packed red cell button at the bottom and a clear liquid layer on top. Carefully aspirate all of the clear supernatant out of the tube without disturbing the red cells. You must repeat this saline wash and aspiration cycle for a total of 3 washes. After the final wash, completely remove the saline, resuspend the packed cells in fresh saline to create a 3 to 5% cell suspension. This washing process removes any residual plasma proteins floating around the cells. If you fail to wash away these unbound proteins, they will neutralize your HG reagent later in the test, causing a false negative result. Perfectly washed donor cells provide a clean, uncontaminated slate. This state is strictly required before we can introduce the patient's serum and look for reacting antibodies.
We are now ready to perform the broad spectrum compatibility test, which occurs in 3 distinct phases. Phase 1.
Clearly label 2 test tubes as tube 1 and tube 2. Add 2 drops of 5% donor cell suspension and 2 drops of the patient's serum to both tubes. Next, add 2 drops of 22% bovine albumin strictly to tube 1. This enhancement media works to reduce the natural electrical repulsion between the red blood cells. Centrifuge both tubes immediately. This immediate spin forces the red cells physically closer together to reveal any large pre-existing cold-reacting IgM antibodies in the patient's serum. As seen in this illustration, an IgM antibody has a large circular pentameric structure held together by a J-chain. Because it's so large, it has a wide enough reach to easily bridge the gap between the red blood cells on its own at room temperature. Remove the tube from the centrifuge and gently tilt it against a well-lit white background. Look closely at the cell button as it dislodges, checking for any visible clumping or agglutination. If the cells flow smoothly without clumping, the result is negative. But this only clears the donor unit of cold-reacting IgM threats. The testing has to escalate to uncover smaller hidden warm-reacting antibodies.
Phase 2.
Place both tube 1 and tube 2 into a 37 degrees Celsius water bath for strict 15-minute incubation period. The 37 degree environment stimulates human body temperature. This creates the exact conditions required for dangerous warm-reacting IgG antibodies to physically attach themselves to the donor cells. This diagram shows the simple Y-shaped monomer structure of the IgG antibody. Unlike the massive IgM pentamer, IgG has a limited reach. It is simply too small to span the distance between the red blood cells and cause visible clumping on its own during a spin. Once the 15 minutes have passed, remove the tubes and centrifuge them again. Gently tilt them against your white background to read the results. If there is still no visible reaction, the cells may be coated in IgG antibodies, but they are not clumped. Tube 2 must proceed to the final chemical bridging phase to reveal them.
Phase 3.
Before adding our final reagent, you must wash the cells in tube 2 3 more times with fresh saline. Centrifuge and completely discard the saline after each wash, leaving a dry cell button. Add 2 drops of anti-human globulin, or AHG, the reagent directly to the freshly washed cell button. Mix the tube thoroughly and centrifuge it for 1 minute. This schematic demonstrates how the AHG reagent works. The AHG molecules act as physical bridges. They grab onto those tiny, visible IgG antibodies, coating the red blood cells, pulling them together, and forcing them into a visible clump. Next, it would be to take the tube out and to check.
Following centrifugation, we gently dislodge the cell button to read our results.
A smooth suspension means comparable, visible red clumps means incompatible, or clear hemolyzed liquid means incompatible. If the result appears negative and the blood seems compatible, the procedure is not over. You must validate this reading by adding 1 drop of IgG-coated Coombs control cells to the tube. Centrifuge this quality control tube because these check cells are pre-coated with IgG. The active anti-human globulin already in the tube will grab them, which must result in forced visible agglutination. Seeing this forced agglutination is your absolute proof that the AHG reagent was active and not neutralized by stray proteins. It confirms your wash technique was flawless, validating the entire crossmatch as accurate. A negative crossmatch reading, paired with a positive quality control check, officially designates the donor unit as serologically compatible. It is safe for the patient to receive. We reach this point through a strict sequence of elimination. We washed out plasma interference, checked for large IgM in the protein phase, incubated for smaller IgG in the thermophase, and revealed hidden threats using HG. Now, finalize the process in the lab, officially label the cleared donor unit with the compatibility results, and update the patient's medical file.
The final step is the accurate documentation and reporting of our results.
Now we must evaluate the strength of this clumping. We grade the reaction on a strict 0 to 4 plus scale. A 4 plus reaction appears as a single solid cell mass. A 0 grade shows completely free unagglutinated cells dispersing into a turbid background. And the reaction between 1 plus and 4 plus or any sign of hemolysis indicates an incompatible crossmatch. This forces the lab to reject donor unit. A completed cross-match is only useful if it is supported by an impeccable chain of documentation. The technician must immediately, legibly record the reaction grades from every phase of testing onto the patient's permanent medical record. We fill out the compatibility tie tag with the recipient's unique identifiers and the donor unit number. This tag must be securely and permanently attached directly to the cleared donor blood bag. Before releasing the blood bag, we perform a vinyl visual inspection of the unit, checking for clots, discoloration, or signs of bacterial contamination. This exact alignment of serological testing, visual inspection, and clerical verification is the absolute standard required to clear a unit for issue, ensuring the highest level of safety for the patient.
By systematically following these procedures, from strict specimen labeling and ABO verification to the three-phase major cross-match and rigorous documentation, we ensure the highest standard of blood safety for our patients. Thank you for watching.